So let's talk about estimating populations using the Lincoln Index. This is the syllabus objective. It's quite simply to be able to use the Lincoln Index, and this is the formula, to estimate the size of a population. Okay, so why do we estimate populations instead of counting every organism and being accurate? Well, first of all, it's generally not possible to count every organism. And that's the whole idea of sampling, which we're going to talk about later. But we're talking specifically here about motile organisms. That means organisms that can move. So it's very difficult. They won't stay still to be counted. And it's particularly difficult for organisms that are small, really fast, or hidden. You know, just like fish, for example, it's very difficult to be able to get them to stand still to be able to count them. So we need to have a way of doing an estimate. And the way we do that is using a technique called mark and recapture. It's a scientific method that we can use to estimate the population of motile organisms. So let's talk about the actual technique of doing the mark and recapture. So essentially, the ecologists go out to their ecosystem and they use traps or whatever to capture a sample of organisms. They count how many they've got and then they mark them. They put some sort of marker on them so that they can identify them again. They release them back into the wild. After a time, they come back and they capture a second sample. Now they count how many have got the second sample and they count how many are marked. And then they can use that using Lincoln Index to estimate the population. So here's the formula for Lincoln Index. The capital N is the estimated population. The capital M is the number marked in the first sample. The lowercase n is the number caught in the second sample. And the lowercase m is the number marked in the second sample. Now that seems like a lot. And at the end of the day, in the external exam, they will give you the formula, but they won't tell you what each of these means. So you need a way of remembering it. This is what I think is the best way to remember it. Think about the M's, this M and this M. The M stands for marked. Okay, so it's numbers that are marked. Now, the capital M is the first sample, and we remember that because sentences always start with a capital letter. So we know that this capital M is the number marked the first time. The lowercase m is the number marked the second time. And then the only other thing you need to remember is the lowercase n is the total number caught the second time. So let's look at putting that into practice. So we've got crabs here we're trying to estimate the population of crabs. So in the first sample, we caught 40 crabs, which were marked. And you can see how they marked the crabs here. Okay, they released them, and the second time, 50 crabs were caught, and 10 were marked. Okay, so let's see if we can remember which one is which here. 40 crabs were captured and marked the first time, so that's the capital M. 10 were marked the second time, that's the lowercase m. And the lowercase n is the 50 crabs were captured in the second time. There we go. And we put that into our formula, and we estimate the population is 200 crabs. Now, there is some assumptions with this. We need to firstly assume that the marks don't actually affect the survival or the behavior of the animals. We don't want the animals to avoid capture, to be resistant to being captured a second time. And we also don't want it to affect their ability to survive. We need to assume that there's no births, deaths, or migration in the population. Now that's quite a limitation really, isn't it? Because clearly that always happens. But we're assuming that the population is closed. We obviously assume that the mark remains visible and that we can identify that in the future if we recapture that organism. We also need to assume that the organisms that we've marked can mix randomly with the unmarked organisms, and that every individual, marked and unmarked, has an equal chance of being recaptured. So look, you don't need to memorize these at all, but I just want you to think about these are, I guess, limitations of the technique. It is only an estimate because it could be biased by for example, putting on a mark that actually affects the survival of the behaviour. We're making an assumption that's not true, and that is that there's no births, deaths, migrations, etc. And we may actually bias it by having a mark on these organisms and changing their behaviour such that they don't want to mix with the unmarked, or the unmarked don't want to mix with the marked. So these are biases we could introduce. Now, so there's some limitations, of course, as well. The marks may fade or get lost. They might fall off, and we can't tell if we've actually caught this one before. Yet the organisms may avoid being recaptured because of the negative experience. Now, a small sample size can really distort the results. I want you to consider what would happen if we have no marked organisms in our second sample. So what would that look like? So we can't divide by 0, so we just can't mathematically do that. So if we couldn't catch any the second time, it means that we can't actually estimate the population. The marks could wear off, and we might not be able to find the ones that we've caught before. The marked organisms might have died, evaded capture, that they don't mix randomly, or it might mean that there's a huge population. So look, there is limitations with this technique. We do need to be able to think about the limitations and the biases that we might introduce, but be aware that it is a technique that we can use with motile organisms.